afternoon, and welcome to the Simon's Electron Microscopy Center's Winter EM course. Today, I have the pleasure of introducing you. I have view of camera, sorry. I have the pleasure of introducing you to Israel Fernandez. He's hailing from the LMB. He previously was working with Venki over there. And I will say that we have a lot of experts. A lot of people are interested in single particle. Please do follow up with the Q&A, because if you are doing single particle projects, these lecturers have a wealth of experience. I will have to say that the ribosome field, one of the fields that Israel is working on, is a very mature field, and it's one where a lot of EM has come together to really push the field forward. Some of the most beautiful gold grids I've seen is coming from Israel's hands, and he can really do things where if you have 7 conformations in there, even if they're minor, he can pick them out. So even though they're like 1 or 5%, I mean, it takes a lot of work, takes a lot of statistics, but that's where you can actually see the whole pathway. And that's something that's really exciting. You hear a lot about, oh, EM can handle single particles, it can handle heterogeneity, but the exciting thing is that it can answer biology, and one can really tease out a conformational pathway, even if it's a minor pathway. So without further ado, Israel Fernandez. Long press. Green, good.
Okay. Thank you so much, Ed. That was a very nice introduction. Yeah, I would like to talk to you about my work at, when I was working in Biochemical and Christian's lab. That was my postdoc, almost 7 years working there, mainly with ribosomes. And as Ed said, it's a very good sample for EM. It's a very rocky molecule, so the grids always behave very well. And that allowed us to do a lot of stuff. So not only solve the structure, but as Ed's pointed out very well, we were able to understand in a structural perspective a lot of the biology going back to this kind of sample. So I would like to give you a little bit of an overview of the technique itself, just in case you are not so familiarized with it. Then I will do a little bit of an introduction about the biology and the biological questions that we were actually interested in making stuff. And then finally I will give you an overview of the data processing that we used to use to identify these all different classes and all these conformations. I think this is not working too well. Maybe from this side.
Yes.
Okay, maybe now. Better?
Yes. Good.
Okay, so just very quickly about single particle cryo-EM. We just need to some purified samples, you know, the degree of purification. The better you have, the most pure the sample, the better. But you know, I'm basically an X-ray crystallography that becomes familiarized with cryo-EM, and the way we purify ribosomes is a very straightforward way. But sometimes you have some contamination that you can survive with in EM. In crystallography, that could be a limitation. But basically, we need to have like a layer of ice where we have our ribosomes. Sometimes we use a carbon support or other kind of support. But lately, I've been dealing with all these gold grids, and we are able to tilt a little bit. So I will talk to you a little bit more about that. But anyhow, we just go into the microscope, we get this 2-dimensional projection of 3-dimensional object like the ribosome, doing some computation, and we get an EM map that we can interpret, hopefully, in high resolution features with a model that we will refine with the stereochemical restraints that I will talk about. Because this step is very important, especially if you have high resolution. So basically, in the last years, you know, we have this fantastic Titan crisis. We have software. I was using EPU when I was at the LMB. Now I'm very happy using Legionel. But basically, you can collect in automatic mode a lot of data, and especially with the ribosome grids that are. Is that me?
I think you're a little bit louder for the mic.
Okay. So you can collect a lot of data from a single grid, and this allows you to actually work a lot. Also, you know, we have detectors here at the NYSBC. We mainly use the K2, but we were using the Falcon 3 at the LMB in integration mode. The time for sample like the ribosome is quite good, because the ribosome is quite good, so it's quite big, sorry. So then the alignment is not an issue, and the Falcon can expose in 1 second, and you have a lot of information and a lot of images per second. But and finally, all the work I'm going to show you today, it has been processed with Reliant, and that's basically the software that I'm more used to to use, especially this three-dimensional classification and some mass classification I'm going to talk about, because it's a novel feature that is extremely useful for the kind of things we were doing. So this is a very brief introduction, and now I want to tell you a little bit about biology, because the whole point for us to start using EM was to understand the biology of the eukaryotic ribosome in different stages. And for that, you have to understand very basic principles of how a ribosome works. And obviously, we can start from a simple diagram of how the genetic information flows from the DNA. Then there is a copy of RNA that actually has to be read to produce proteins. And this is a kind of a very interesting point, because the ribosome has to be able to identify what is called initiation codon, which is always an AUG, and how this is identified and located is actually setting up the reading frame, and it's absolutely essential for the cell to do it properly. So once the ribosome identifies the AUG, he places a very specific tRNA in that position, base pair, with Waxman and Crick geometry of, as I said, a very specific initiation tRNA. So the delivery of this first tRNA to the initiation codon is an absolutely essential step in any genetic transmission information from DNA. And this is done in several ways in different organisms. In bacteria, the ribosome uses two very small protein factors, which are called IF1 and IF3, that basically block the A and the E side, so the P side can recognize the initiation codon, and the tRNA by itself find its way. It's very important to realize that in bacteria, this has no work. In bacteria, you have this physical interaction between the RNA of the ribosome and the messenger RNA. There is a physical recognition of a short sequence of bases called the Shiner-Gano sequence, and by just doing this pairing, the initiation codon is quite close to the P side. So the ribosome, with this little information in bacteria, that's important, he's able to place the right tRNA in the right place. In eukaryotes, however, things are much more complicated. There is some similarities because there are two small proteins that are homologs of IF1 and IF3, but that's it. In eukaryotes, we don't have physical interaction between the messenger and the ribosome, and the messengers in eukaryotes are modified chemically on the 5' with this modification, which is called the cap. This modification is actually it's very important, as I will tell you later. But the main difference is that the tRNA, the initiation tRNA, the first tRNA, is delivered to the ribosome with a specific set of factors and GTP consumption. So this process is much more complicated than in bacteria. Then you have a set of proteins that actually recognize the cap, and with ATP consumption, look for the initiation codon. This process is very dynamic and extremely flexible. And finally, we have an initiation factor which is gigantic, as big as the small subunit, which is called EIF3, composed of 6 subunits, 6 peptides in Gs and like 12 nanos, that basically interact with every single factor at the same time in different conformations. So if you think about this, we were able, I mean the ribosome community, especially Benki, in his group, they were managed to crystallize several conformations of these guys. This is a nightmare for any crystallographer because it's extremely big, extremely flexible, and with a compositional heterogeneity. That's why nobody have any information about this. And now with the new capabilities of EM, we are actually able to see something. So after all this has happened, the first tRNA is in the P side. Then there is a last step, which is basically the checking step. And here the two kingdoms of bacteria and eukaryote basically converge, and there is a protein which is called IF2 in bacteria and an homolog in eukaryote which is called EF5B, that with GTP consumption check that the tRNA is delivered in the right place in the right moment. Once this GTP is hydrolyzed, the ribosome assumes that the initiation has finished, and then it starts the elongation in a very rapid fashion. So the last thing I want to tell about this biological introduction is that this is important because many viruses tinker with this regulation to produce their own proteins. Viruses are obviously devoid of any ribosome, so they need to hijack the ribosome from the cells to be able to produce their own proteins. And this hijacking is basically executed at this level, at the initiation level. So they have several strategies. There are many different virus strategies to kidnap the ribosomes. But one which is very interesting to me is that they are able to have a very specific folded RNA sequences in the messenger itself to be able to mimic some of the initiation factors. And this is what I'm going to talk about later. So how we decided to tackle all these problems? Basically, we were trying to isolate first very high quality ribosome for EM. And we were looking for several species. At that time, we were a little bit biased by crystallography, because in crystallography, you always try to isolate the very pure, very homogeneous samples, otherwise nothing will crystallize. So we were screening several species of yeast to be able to isolate ribosomes. And as I said before, the purification is a very straightforward way, kind of tricky, but anyhow, we use a lot of sucrose gradient, we use gel filtrations, to be able to first check that we have all the ribosomal proteins, large subunit, small subunit, and then once we have the ribosome purified, we check them on a native gel to be able to see a single bond, so everything is running very nicely. Once you have this, oh, sorry, I forgot about this. So this, we tried many different things at the time, and one of the things we tried, we isolated ribosome from a thermophilic algae from Japan, I don't remember the name. It was really problematic because it was blue, and we could only identify the ribosome in this kind of band that was always overlapping with this pigment, but we were able to find the right ribosome once we changed the concentration of salt, so the pigment was somehow washed away. But anyhow, the whole point of this diagram was that we were looking for a lot of different species to be able to find ribosomes that were very, very stable, so we can reach high resolution by nowadays, the technology, as Ed said before, has moved so fast that almost, I mean, you can have high resolution with human ribosomes. So all this doesn't make sense anymore. But yeah, I just want to tell you because it was a lot of work. So the idea that we have, once we got a very nice ribosome from this organism, which is called Cryo-EM lactis, is a very simple yeast that actually has a very rocky ribosomes. We were very happy with these bands. We were thinking to program the ribosome in specific stages. It was not just looking at the ribosome. We want to have the ribosome in a specific conformation and in specific stages, so we can actually see in a structural perspective, within a structural perspective, the shape of the whole complex. So for that, to be able to program a ribosome in a specific stage, you need some kind of biochemical tool to tell you that you are in the right track. So given that the ribosomes are so big compared with the proteins that actually interact with, we were using what is called a sucrose pelleting assay, which is a very simple assay, but it's extremely useful. So basically, you have, this is a centrifuge tube, where you have a thick layer of 1 molar sucrose, what is called a cushion, and then you have some buffer where your protein and your ribosome are happy with on top of that. So basically, you mix your ribosome with your protein. This is not at scale, of course. The ribosome would be like a building here. But then the idea is that you get these guys together in the buffer where they are happy, then you centrifuge very, very fast. And given that the ribosome is so big, he can go through the cushion and pellet, while the protein by itself, or the factor that is actually interacting with the ribosome, if it's not bind to the ribosome, it will remain in the soluble fraction, you know? So again, if things are interacting, the ribosome basically tracks with him wherever he's there. But if the ribosome is not interacting, basically you get this guy in the soluble fraction, you remove it, and in the pellet you only have ribosomes. So just to give you an example of how this biochemical test works, here there's this protein called Relay, which is it's another project, but it's basically a very nice pelleting assay that I did with this project. So this is the purified protein by itself. And this is just ribosomes, 70S bacterial ribosome, mixed with the protein in the absence of any tRNA or mRNA. The whole idea of this project is to prove which requirements in terms of tRNA this protein have to bind to the ribosome. Okay? So in this lane, you see that there is a little bit of pelleting, but very little. When you add a little bit of mRNA, nothing happened. You add a full mRNA, nothing happened. But then when you have 2 tRNA, sorry, 1 tRNA, things start happening. And when you have 2 tRNAs, all ribosomes saturated with tRNA, you have a stoichiometric pellet of the protein. So this is telling us that actually this protein requires 2 tRNAs on the ribosome in a specific position, because we can deliver tRNAs to the A or the P to get the pellet, to get the binding. And actually, there was a kind of an indication that there is one domain on the C terminal that binds to the ribosome. We make a mutant without this domain, and when you get this, there is no pelleting at all, even in the presence of tRNAs. So these kind of very simple experiments help you out to narrow what part of the proteins are interacting with the ribosome and in which conditions. So once you get this very nice skeleton condition, you can go to the structural characterization. How do we do this? We normally, at the LMB here we have an F20, but at the LMB I did all my optimization with a T12 microscope. A T12 microscope. So, yes?
Can I ask you, what size do you think, what is the smallest size that such an assay would work with?
The pelleting assay?
Yeah.
Depends on the concentration of the cushion. Depends actually on the percentage of the cushion. Whatever is, I don't know, I would say 1 order of magnitude, maybe. So if you are working with a 100 kilodalton protein, a megadalton thingy, I don't know. So yeah, once you have characterized your interaction, we were working a lot in a spirit, in a microscope, a cube with a cryo condition and a CCD camera where we can do a lot of stuff, just checking the eyes. It was a lot of work. Also, the buffer conditions are very important for the ribosome grids. So a lot of optimization was required to get something like this. This is a ribosome from bacteria in a spirit microscope. Looks very nice. If you increase the magnification, you can see that actually you have a lot of different views and the contact is very, very nice. But also there was a lot of problems. Sometimes you get things like this, you know, bad eyes or things like that, contamination that nobody knew where they're coming from. So anyhow, the whole point is that we spend a lot of time doing optimization, so don't run to the cryos as soon as you see one particle, okay? So these scopes are extremely useful. You get a lot of information. Now in Columbia we have an F20 with a K2, which is a fantastic instrument. We have a lot of information and only go to the high scopes when you are really confident that you can have very good grids. Otherwise, there's no magic on the Titans, you know. If you can see your particles in a small scope, you won't see it on the cryos, okay? And then we used to use a lot the polar microscope with, as I said, with the Falcon 3, and then we also have the Titan cryos, basically like they have here cryos 3. And we normally have like a 1 day collection time over there. Here you have 2. The focus range between 2 and 3. Sometimes you can go 1.5 or even 1 if you are using like a very thin ice, and you know it was a pretty straightforward data collection. So the way we used to do things over there, things have changed since then, but basically, obviously we were using a motion core. We're using CTF fine to correct the CTF. Now I'm using GCTF that I really like. And also the particle picking, it was done in Reliant, but now I'm also using G-Automatch, which is KI software. And a lot of, everything was, as I said before, engineering, analyzing Reliant. So, we used to do a lot of 2D reference-free alignment to check the quality of the data set. And for the ribosome, normally we were able to distinguish at this stage between a full ribosome, large subunit, and a small subunit. Sometimes there were a lot of particles like these guys that they just don't align, probably bad particles or gamma ribosome or whatever. But also at this stage we can clean the data set very well of individual subunits, okay? So we know that our factors only bind to the full ribosome, so these particles are basically individual subunits which are not interesting to us. Also, what we have seen is, this is another one, for example, this is large subunits only. We have the feeling that actually the vitrification step is extremely traumatic for the ribosome. So things that you can see in a biochemical assay, you have 100% of ATS. Then after you do your analysis with to this, you see that maybe you have 30% of free subunits. So something is happening when you freeze things, and that's something you have to bear in mind. Also, it's very important to screen different ice thicknesses because the orientations of the particle change a lot depending on the thickness of the ice, and that was also quite important. But basically, once we get the ATSs, we were able to, the way I used to work is based on what sources used to tell me, is like first we do a full refinement with all the data set. We do what we call a consensus refinement, whatever you're going to tell. And not only that, you start doing classifications. So this is consensus refinement that can go to high resolution, but when you do a slice through the map, you can see that actually there is a lot of heterogeneity. So in this region of the map, you can see very, very well-defined features. The secondary structure, very nice, but this other part of the map is really fuzzy, and that corresponds to the small subunits. So if you look into this, there's some kind of indication that there is a kind of a stuff between the two subunits, what's called intersubunits in space, and that was what we were looking for. Just to make things clear, this is the 60S in blue, this is the 40S in yellow. So after a lot of 3D classification, we were able to actually improve the resolution of the small subunits quite dramatically. And this has to do with the fact that the 40S is quite flexible. It's a very flexible part of the complex, and we have several conformations. That work was done using the classical 3D classification in Reliant, but now, as I will tell you later, we have these masking methods with signal subtraction that is working extremely well for me. More details later. So as you see, that different when you are able to classify this, suddenly the density for this guy in the middle seems to be better, and and also you can even mask out the whole 40S and forget about the large 60S, but you pay a price, which is losing the resolution in that area. And one of the problems we have with the ribosome maps is that they are extremely heterogeneous in density, in resolution. Some areas are really good, as you can see here when it goes through the slice of the map. It goes clearly at 3 angstrom in the core of the ribosome, but areas of the factor, which is here, is clearly beyond 5, 6, 7 in this kind of low resolution, if you want to say it. And this has to do because the ribosome, as I will show you later, it has a lot of degrees of freedom. The small subunit is moved by itself. Several regions move independently of that movement, and then you have this factor that binds, and the binding is never 100%. But as I said, now we have a lot of computational tools to deal with these problems. I just want to tell that actually all the building and the refinement was done with CUT and RefMac in our maps. And there's a very nice paper written by my friend Alan Brown where all these things are very well explained. So if you want to have a look at this paper, I think it's very, very useful. So just very quickly, I want to give you an overview of the kind of things we were doing. So that was the first paper we managed to get at something like 7 angstrom resolution. And it was very important at the moment because it was the first time we were able to see anything eukaryotic. That means it is the first complex of eukaryotic factor with an eukaryotic ribosome, and we were able to see an initiation tRNA, as I said before, positioned by EF5B, which is this GTPase that check that the tRNA is in the right position. And as you can see here, there is a kind of an arm of the EF5B that actually grab the tRNA and check everything is fine. And this interaction is very important to activate the GTPase activity of the protein. We identified this region of the protein, which is universally conserved, and it has to do with, again, this activation of the GTP analysis, of the GTP molecule. Then we applied the same kind of strategy to study one of these viral internal ribosomal entry sites, which are these fragments of RNA which are able to fall by itself and mimic some initiation factors. And we actually could see very nicely how this part of the virus basically blocked the access of the tRNAs to the ribosome to hijack all the ribosome. Also, in the small subunit, we see a lot of factors related with the canonical initiation. And finally, as I said before, all this work was done in GIST, also this one, because we thought that actually it was required very homogeneous samples to be able to reach high resolution. But then working with Rebecca Burgis and Manu Hetz, they purified mammalian ribosomes from pig pancreas, and it was fantastically good too. We reached 3.4 angstrom here. We were absolutely astonished by the quality of these maps. And we can also see actually this factor, the 661, the transport channel, very well defined. So that was basically what I want to tell you about this, but now I want to change to the other more technical talk that I have over here. There you go. So, yeah. So this is this iris region, this iris molecule that I was talking to you about. Sorry, this should be in here. Okay. We were basically trying to understand how this small RNA, which is only 180 nucleotides, is able to block the access of the host tRNAs to the ribosome, move around, and be able to hijack the translation capabilities of the ribosome. There were some indication in the literature that actually there could be some kind of effect related with this green region, which is called the pseudonode 1. But based in our structure, what we can see was something very interesting is that the iris binds directly to the ribosome in the intersubunit space, inserting this PK1 region in the A side, which is the first binding region of the tRNAs on the ribosome, and by doing that, they basically block the access of the endogenous tRNAs to the ribosome. Then he induced a kind of very dynamic movement of the ribosome, mimicking the tRNA movement, to induce a translocation that free the first tRNA and then the the first A side of the ribosome, so the first tRNA can bind, and then there is a second translocation to finally place the tRNA in the right place. So all this is like a little bit too many details, but what I'm going to tell you is that we were able to follow the whole pathway of this iris by Cryo-EM. So we did first this interaction with the ribosome, which is very dynamic, as you can see. So this is the small subunit, this is the large subunit, and that was exactly the slice of the map I showed you earlier. We were able to identify these two classes and characterize this movement that is actually coupling the movement of the small subunits with this region over here, which is called the L1 stop. When you do this, you basically have a kind of a hijack ribosome. We were also interested to know what happened before this happened, how this small molecule is able to bind first to the small subunit and then recruit the 60S to get this kind of dynamic complex. And we were able to do this by Cryo-EM using a purified system with these two guys. And this is what I want to tell you about a little more in detail, that we are using these masking classifications, which are working very well for us. So we start with, as I said before, 2D classifications, then we did 3D classifications to get like a more or less just 4ES particles. And then we did a whole refinement with all the particles that reached 3.7 angstrom, sorry. But clearly the region of the iris was some kind of weak, and that was because there was a mixture of free 40S and 40S with iris. So when you actually go in Chimera and load your maps and go up and down the threshold, and you see that the threshold in one area bumps earlier than the other, it's because there's probably a problem with the occupancy. So what we did was create a mask, very wide mask, like a 25 pixel extended mask of the area of the iris. And then using this signal subtraction in Reliant, we were able to very quickly remove 58% of the particles, so more than half were empty. And this is something you have to bear in mind, because this iris biochemically bind at the picomolar affinity. Yes?
You mask the iris density, or you mask the surrounding density of the ribosome?
So you get a PDB that basically coincides with the shape of the iris, and then extend that 25 pixels out and make a mask out of all this region. Okay? So you basically, in Reliant, you remove all the density outside the mask. Okay? So you can use only the signal inside the mask to do this classification without alignments. Okay? So, as I said, it was very surprising to us that something that binds with picomolar affinity to the 4ES, so many empty 4ESs, because also the excess of the iris related to the 4ES was 10 times. So it was a huge amount of iris. And I think it has to do again with the vitrification process. I think it's very, very dramatic. and basically cracks and kick out all the binding factors. So that's something to think if you are making a complex or something that binds to something that in vitro can look one way, in the grids can be quite different. But again, I think the new classification schemes are working very well. I'm using this signal subtraction in rely on with other complexes. It's kind of tricky, so if anyone wants to have any problem, let me know. I can help you a little bit with a couple of tricks. One trick is how big you make the mask. It's absolutely essential. The other trick is the T parameter that you have to use in rely on. This is extremely heuristic. You have to try different things, but the fact that it's so fast is actually quite helpful. And the other thing is that you need to know a little bit what's going on with your molecule. If you know that this region of this factor binds on alpha helices, maybe you can include this alpha helix. I don't know, try to be imaginative, but again, if anyone have any problem, let me know because we can maybe help. So anyhow, once we get the particles with the iris, refine again, and we do more classification, and suddenly there was still like 30% of particles without the iris. So it was a very pervasive problem, but actually the density, once you have all the particles with the iris, improved dramatically. Okay, so you have 5 or 10% you are looking for a complex between two molecules, and then you have 5 or 10% of molecules without the factor. It's really a problem. So don't think that actually because it looks like you have something, you can always improve it. Yes?
Is it sort of this, can you treat it the same way if you have not, that it's not bound, but that it's in a different conformation? Like, for example, my situation, I have no density for DNA, but probably some of the particles do have it. I'm not sure. I can maybe talk to you afterwards, but you can use masking.
Yeah, and actually what we are doing is, that's a good question, because in another complex that I'm going to show you later, which is more complicated, I have several conformations apart from this. So I have empty, conformation 1, conformation 2. But you can play around with another parameter in Relio, which is the number of classes, which is also key. So it's like the size of the mass, the number of classes, the T parameters, all these things you have to play with, and the results can be really different. And it's quite difficult to judge. You have to look at the maps and you have to try to understand a little bit what's going on. Yeah. Okay. So that's one thing. Then we managed to refine this guy, some kind of high resolution, around 4-ish, more or less. There was a region that binds to the small subunit that was fixed, that was very, very well defined, but another region that was actually not interacting with the air, that was quite flexible. And this is a very important figure in my opinion, because one of the problems the EM people is having is like they don't know what to do with the maps. So you have to generate a good stereochemically refined model. And we have a lot of knowledge from crystallography on how to do this in the best possible way if you have good data, if you have good maps. So in this case, what we like to show, that was Gary Murshidov and Richard Henderson idea, is like you have your half maps comparison here, the Fourier self-correlation, that you always have to match with the model to map Fourier self-correlation of the two half maps. Okay, so here basically there are 3 curves, but you can only see 2 because these 2 are overlapped. They are exactly the same. This is the model versus the map versus half map 1 and versus half map 2, and one of the half map has not been included in the refinement of the model. When these 2 maps are the same, you know that your model is not overfitted, and then you have to have a look to the B factors of the model, of the refined model. When everything makes sense, you can see that actually the refined B factors for the atoms of the model coincide with actually the local resolution estimated in ResMap. So it's important to pay attention to the model when you have high resolution. It's extremely important because there are a lot of tools, a lot of information that can be gathered from crystallography and the refinement of crystallography models. Okay. Anyhow, that was just one thing. So that was the structure of the iris with the Fourier complex only. But as you can see, well, I kind of skipped the biology, but basically what we saw is that actually there is Watson and Crick interaction in the decoding center of the ribosome that the ribosome recognized as a cognate tRNA and mRNA interaction, as expected. But it's a very, very sophisticated way. This mRNA is able to fall into a kind of a like tRNA molecule. Then we wanted to follow up what happened after this dynamic interaction that we described earlier. And what happened is that the iris has to be translocated. So there is a factor, a canonical factor, which is called EIF2, which with TTP consumption is able to move out the iris from the A to the P, and then the first tRNA combine, and then the ribosome is committed to the production of viral proteins. So we wanted to know how this happened, and we just produced in vitro this protein, made the complex, and did a lot of microscopy to be able to get this. So this is again a classification method that we employ to solve this interaction, and this is related to what you were asking earlier. Basically, the strategy was the same. We collect the data set, do declassification, and we jump into 3D refinement of the whole data set. That's what is called the consensus map. And when you do the consensus map, you can have a view of where are the regions that are actually heterogeneous. I like to look at the maps through the Reliant slides. You can see actually the map in different slides, and you can see areas which are really well defined, others not. When you do this, we were able to define a mask that includes all the intersuburate space. It was a very broad mask. And using like 4 or 6 classes, playing around with the T parameter, we were able to basically identify one class, 25% of ribosome with the iris, 42% only EIF2, and 30% both at the same time. So these things can be done very easily in Reliant, again, because if you refine first all the data set, you can use what is called you can skip the alignment, basically. If you do classification without alignment and signal subtraction, you are able to have these things in the ribosome very easily. But the problem again is like how to define the mask and how to define how many classes you want to use. And it's tricky. That's the only thing I can say. And finally, once you have the particles with the 2 factors, we'll still be able to distinguish 2 classes. That was with the 2 factor and different conformation of the small subunit. So the degree of heterogeneity was fantastic. You have like 1, 2, 3, 4, 5 classes in the same sample. But it's not a problem in the ribosome because, as I said, it's very big, it's very well aligned, and with 20,000, 25,000, maybe 30,000 very good particles, you can go high res, no problem. So again, I just wanted to show you these kind of graphs that I like a lot to show. This is the local resolution for the iris with this factor, and this is the refined factor again, not overfitted, and you can see the V factor are actually coincide very well with the local resolution area. So that was something nice. So finally, this is the structure. It was something like 3.6. Very well defined areas of this interaction region that was very useful for us. But I think I have a movie that I did. There you go. Let's see if it works. So this is just morphing between different stages. So this is how the iris bind. Then EIF2 basically push out the PK1 from the A side. And that was very interesting to see, because when the canonical tRNAs move from the A to the P to the E, they go in what is called hybrid states. They basically bind, tilt, move, bind, tilt, move. And what we saw is actually the PK1, which mimics tRNA, also mimic this kind of hybrid states. So it's a pretty remarkable molecule. 45 minutes, I think I'm done. That's the last slide, that we can actually see the whole cycle of how this iris is able to hijack the ribosomes. And I just want to quickly acknowledge the people that helped me out a lot at the LB. Of course, my boss, fantastic boss. We did the second work, the EF2 work with Jason Morrow, a guy from Texas, very cool guy. Alan Brown helped a lot with the model building, and then all Richard Henderson people, especially Chris Russo, I worked with a lot, Gary Mursilo, Paul Emsley, Christos Sala, it was a really nice interaction there. And of course, Sorceress is a very good friend and extremely clever guy. Thank you.
So we're trying to leave enough time for more Q&A, because I think we feel the single particle section is the most interesting based upon your surveys that you want to know more about. So, how many people are doing single particle right now? It should be over half the class. Yes. Okay, so in terms of these first couple of lectures, have they been informative? What do you feel is missing? What do you want to know to help push your research?
For me, I'm just starting out. That slide you had with the workflow is probably the most helpful.
Where you see like CTF,
Yeah,
like what
starting with the
particle
raw
picking.
images, what programs you're using, what data you take from, in each step, then just throwing out junk, whether you're keeping the entire data set, all of it, that's just very helpful.
There are several options. I mean, it depends on your preference, different software. As I said, now we are using, I guess I'm at the NYS, you see a lot now, so I really like what these guys use. So I think you have to be open mind to try different things. The field is moving very fast. There is always new stuff, but at that moment we were using Motion Core 1. Now you use Motion Core 2, works very well. Other people use Amblur from Nico Grigoriev. Other people use the Spanish software, XMIP. I don't know, just try out things. I'm using Motion Core 2, now GCTF, because it works very fast in my hands. And also one of the good things of GCTF is that you can do this local CTF correction with the particles, which is very, very good in the ribosome, because the ribosomes are very contrasty, so the signal can be estimated very well. If it's more smaller stuff, I'm not sure it's going to work so well. But with you have a big particle, GCTF with the local estimation is extremely good. It's slower though. Also, the particle picking, the Reliant tool is okay, but I mean, these guys use another one. I use G-Automatch now, which again is very useful, especially if you combine with GCTF. And again, Reliant is working very well for me, but now I'm using also Cryo-EM. I'm trying to do CSTM, so I don't know. I think it has to do more with the detectors. Almost everything works if you know how to use it. So that's my opinion. But also, I think it's very important. And one of the things we are having problems in Colombia that we have a lot of new users is they have this, sorry to say this, a very American mindset. Just get numbers, oh, my finger's not working, then I need 2 million particles. It's not really like that. You need very good particles, and if you have 50,000 good particles, you are in a good way, in a good position. With the ribosome, of course, but with the smaller things also. So I think it's better to spend a lot of time in the optimization stage, spending a lot of time with a small microscope, trying different grids, trying different methods to get the best possible grids before going to the big scopes. Because otherwise you're going to waste money, you're going to waste time, and you're going to piss off these guys. So that's my and also another thing that I spend a lot of time, I would say, in my data processing, normal way of working, I would say I spend 60% of the time in the classifications. It's really, really important. And as I said before, it's better to have a very good small set, very well classified. It's more informative a 5 angstrom map with a very good classifier than a high resolution map with different regions which doesn't look well at all. So don't worry too much about the Fourier self-correlation number. You have to look at the maps and make sure that they look better. Because the images and the detectors are so good that a 4.5 angstrom map looks so much better than a 4.5 angstrom map in crystallography. It's really informative, so that's my advice.
Yeah, you mentioned the number of classes that you pick for 3D refinement is really important. Maybe you could expand on that.
Yeah. So in my experience, and also talking with Source, is it's important to allow Reliant to have a space to move. For example, in this case, if I will have said 3 classes, he will be completely confused because he tried to fit things differently. So my approach is give the number of classes that give you at least one empty. And it's the same for the 2Ds. If you give a lot of classes, then if you give very few classes, he just tried to fit everything wherever he got into there, and everything is messed up. If you give more classes, he has a space to move around, and when there is one class which is empty, I'm happy. If not, I repeat with a couple of classes more. That's my opinion, and also it's very important the T parameter. It's the only parameter that you have to explore in Reliance, so it's not that bad. The smaller the thing, the higher you have to go in general. With ribosome, we use T parameters between 2 and 6. But my other stuff, which is not ribosome now, that is something around 220 kilodaltons, I have to go to 30, 40 in the classification, so just play around with it. But in general, just going back to your question, it's like just give enough classes to have at least one empty. That's what I'm doing.
It's a lot better now with GPUs because back when we were on one workstation and CPUs, it's painful. It's like, okay, let me try this. Like, oh, not enough. Let me try again. And then a week later, oh, oops.
Yeah.
Now it's a few hours. It's like, oh, okay, now you can play around.
So at the time, we have a cluster of 1,500 CPUs at the LMB, and this processing took weeks. Weeks. Now I have a box with 4 GPUs and 16 CPUs, and I do this classification with double amount of data. I can do 3 or 4 a day of these local classifications. So again, spend time in the classifications because it's really worth it. It's really worth it.
That underscores how fast the single particle field is moving. Not just hardware, but software is the real development over the past few years.
Yeah. And also, I have to be honest, I'm a big fan of Source, but I'm very impressed with Cryo-Spark. Cryo-Spark has now this kind of capability, which is called ab initio, which basically takes the particles and generate models. So you can start like a kind of ab initio classification if you give like a 4 or 6 classes. And for one of my data sets, which is not ribosome, it was absolutely fantastic. It also has this other capability, which is called heterogeneous refinement, which is more or less like a class 3D. But again, just play around with your data and different software and try to figure out what is working for you. And also, you can contact the developers. Are always people really helpful. So maybe because Source is my friend, he always replies.
Any problems with making grids?
Anyone using gold grids? Which hole size?
The hole is like 1.3.
1.2, 1.3?
That's something, in my opinion, for ribosome, everything works. As I said, you can use any grids, it's going to be fantastic. But with other samples, I've realized that actually the grid you choose is absolutely essential. Can be a very big difference. Actually, in one of the complexes I work with one of my students, it's like I don't know why, it has extreme affinity for carbon. So if you just use carbon grid, you don't have anything in the holes. So with these gold grids, we were able to see it on the ice for the first time. Also now, if you have problems with the grids, you can use the Sporidome here, which is amazing. But in general, spending time with the preparation of the grids is very important. Actually. Also the buffer, try to link the biochemistry of your sample with what you see in the microscope. For the ribosome, it's essential to have a specific concentration of magnesium and salt. The ratio between monovalent and divalent agent is very important to have a very stable complex. And that's something you can play around with in the grids, try to figure out which is the best condition for your sample and try to use it on the grids, if possible. But another thing you can try if you have problems with the stability of your sample or aggregation is using a little bit of detergent on your sample. That also helps a lot. There's a lot of things to try, but again, all these things has to be tried in the screening process. Don't think just because one day you see one grid, one particle good on the spirit, you just have to go to the Titan. You have to be able to reliable have a lot of particles on your image that looks nice. That's the criteria. And if I were you, I would just try to get the initial data set in a like an F20 or something like that, because the 2Ds are very informative to know if things are working. That's the way we work in Columbia now. We have a F20 with a K2 that we collect maybe in a day 100, 200 images, and that's enough to actually do some nice 2Ds and be able to judge the quality of the sample. So, for example, Sasha Sobolewski's people, they are working with this membrane protein in nanodisk, which is a very complicated sample, and it's they basically were able to have like 3 or 4 grids in the same day, 50 images each in different detergents, do the 2Ds, and you can see a huge difference just by the detergent you are using. No? So this is the kind of things I will spend a lot of time.
Any other questions? Anyone working on complexes? Your complexes are all stable. One confirmation, everything's great. Great, okay.
I have another question. So, our complex like has this kind of behavior that people have said seems kind of odd, where they seem to coalesce in the center where the ice is the thinnest.
Okay.
And I'm wondering if you've ever seen this kind of behavior.
That's good. So it's broken in thin ice. That's exactly happening to me. Yeah. So I have a very I have the images here. No. I have a complex that actually we thought that everything was done. So it's a complex of 3 proteins, a small protein and 1 non-coding RNA. And biochemically, it looks very, very good. It's a single peak in the gel filtration, everything makes sense. But when we went to the grids, we just take images in the center of the hole, and everything was like a disassembled, like a really ugly. And then one day, we just take an image at the edge of the hole, and there was beautiful particles all over the edge. That was using gold grids. This is this complex that attached to the carbon, using gold grids of 1.2. So what is happening is actually that you have a gradient of the ice on the holes. In this case, if this is the hole, your ice is basically doing this, no? So here you sample, disassemble, and here the ice is thicker, so we could see very well. Those were using 1.2, 1.3 gold grids, okay? So what we did is just use the 0.6, which is basically half of this. And when we did this, actually this gradient was far less, and that was really, really good. So that we're using 0.61 gold grids. And what we are trying to do now is generate the 0.52 grids where the holes are going to be even smaller. Okay? So this is one solution. The other solution is try to get some support. Carbon is a pain because you lose contrast, but lately I'm using this graphene oxide thingy, which is working quite well for me. Just play around with the grids. If you can see that actually your particles looks better at the edge, just use the smaller grids, or just collect close to the edge. You can do this with Legend very easily.
Well, if you have any more questions, you could always email us. We'll forward them to Israel.
Yeah.
And he'll supply a copy of his presentation. You can see it online. And we'll see you when? There's nothing Wednesday?
There's no recitation this Wednesday, so it'll be next. So we'll see you next week. And the homework will be for the practical will be due next week as well.
Okay. Well, then we'll see you Monday.
Thank you.
Thank you, Israel, one more time.
You understand? Yeah.
Thank you. That went well. You can give us a PDF of whatever you can